All right, thank you very much for inviting me here. It's a real pleasure to come out and talk with you all, and also to get away from DC for a week, given the things that are going on there. Whatever changes you made, it will no longer advance. So I will just try and push the button, which no longer works. I'm going to talk about this. This is a picture from Mongolia. Well, you figure that out. And these are Mongolian gazelles. And what did you do?
I just clicked on
Where
the
did you
screen,
click?
on the screen, and then it works. Do you want to try that again?
All right, we're good.
Yeah.
All right, thank you. All right, so today's talk is about home ranges. I'm going to give you a little bit of definition and some perspectives, and then I'm going to talk about the work that we've been doing in my lab and with my collaborators over the last 10 years, focusing on a continuous time stochastic process approach to home ranges. And now the last part of the talk will deal with some applications and recent advances. And so when we talk about home range in the group that I work with, we're using Burke's definition from the Journal of Mammalogy, focusing on the area traversed by an individual in its normal activities. And this is going to exclude, random wandering, exploratory visits, checking out the neighbors kind of thing. And so this is the framework in which the mathematics that I'll be discussing and the interpretation that I'll be doing operates. Now, home range is a subject that has had extensive inquiry. There's been a variety of different approaches that have been used to ask these kinds of questions. How big is an animal's home range? Where is it located? What governs the size and shape of the home range? I'm not going to go into all of these things, in large part because there's an expert already in the room, and he literally wrote the book on the topic. What Mark's work in mechanistic home range analysis is focused on the formation and change of home ranges, and it's based on the correlated random walk framework, trying to encapsulate movement behavior in these mathematical descriptions, which can then be scaled up to characterize the structure and change and operation of home ranges. I'm not going to do that. I'm talking a different approach. What we're doing is a purely statistical approach, trying to characterize home ranges, because people that we work with, the people that are collecting the data in the field, they're often interested in asking questions with relevance to a particular applied question. They want to know how the home ranges are formed and how big they are, where they're located, and oftentimes this is connected to a specific conservation question. And so we're focused on this question of just characterizing, using mathematics and statistical approaches, the size of home ranges and their extents. And so there's a couple big categories of approaches which have been used historically in this subject area of statistical characterization of home ranges. Just very qualitatively, they come in two flavors. One is focusing on the convex hull methods and varieties thereof, which are basically, in a statistical sense, putting bounds on things, or putting a spatial limit on the extent of data. And there's techniques like minimum convex polygons, or more other techniques called local convex hulls, which others have developed and applied to this question. All of them have this flavor of trying to put a bound on something. Another very different flavor is focused on kernel density estimation, which is basically trying to build up a distribution based upon smoothing underlying data points. And so kernel density estimation actually has a very long and formal theory in statistics, and it has found widespread applications, not just in ecology, but very broadly. So if you're not familiar with this, a very simple example, in one dimension, you have a whole bunch of data points shown in red, and you're trying to understand the distribution of those data points. And the simplest way you can do this is to build a histogram. But beyond that, you can also use this kernel density estimation to build a distribution around these data points, which you can then see is a reasonable approximation for the true distribution in green, which gave rise to those points as they were sampled. And so in one dimension, you can see this kind of thing, but in two dimensions, the kernel density estimation is better visualized like this, where you might have a collection of data points on some two-dimensional landscape. And this smoothing process works by putting down a kernel, like you've probably seen in integral difference kernels, but here it's a probabilistic kernel that is centered at each of the different sample points, and the diameter of that kernel is called the bandwidth. So this is it's actually technically it's a covariance measure that goes into putting probability mass associated with each of the data points in one or two dimensional space. And what I'm going to be talking about today is an extension of these ideas and their application to animal movement research. As I said, a variety of statistical approaches have been developed and applied over decades. People in ecology have used a tremendous array of different approaches to try to understand and quantify how big the home range of the particular individuals that they're interested in are. And if you want to get a sense of how these different approaches work together, I would point you to two very large syntheses that were led by Mike Noonan, who is a Canadian based here at University of British Columbia in Okanagan. He had a very interesting history. He was a Rhodes Scholar and a Smithsonian postdoctoral fellow, two very different areas of superlative performance. And he led these two large-scale syntheses comparing, and from a statistical standpoint, the performance of a variety of different estimators for home ranges. One of the conclusions coming out of this is that the technique that I'll be talking about has superior performance qualities in areas of statistical estimation of home range, and I'll be explaining why that is. But rather than go into the full scope of the comparison, I'd simply refer you to these two comprehensive analyses. And so what I'm going to talk about today is called autocorrelated kernel density estimation. I need to give you a background of what it is from a mathematical standpoint and where it came from. I'm also going to give a shameless plug for an R package. I did not create this R package, but the people that I work with did. So I'm not taking credit for it. I'm simply pointing out they've done a really good job, and you can look at this as a resource. But the issue is that modern data from animal tracking studies, which are increasing in numbers and diversity as a way of getting information about wild animals doing the things that they do in nature, has a particular characteristic to it, which is that the data that you get out of these tracking systems are heavily autocorrelated. So this is a picture of the frequency of different studies or different time series in which animal tracks have been analyzed in terms of the degree of autocorrelation they have at the one-time step scale. And these are other papers which have looked at autocorrelation in the past and deemed autocorrelation not to have been an important consideration or something that could simply be dealt with in a variety of ways that did not actually embrace or quantify the autocorrelation that was present. Basically, you could do things like thinning the data to get rid of the autocorrelation signal and therefore work with the modified data set so you don't have to worry about autocorrelation. The problem is that when you look at animal moving tracking data, the amount of autocorrelation at a single time lag is over here at the far right-hand side. Almost all animal tracking data sets that we analyze at this time show a tremendous level of autocorrelation at this one time lag, even though these other studies, which deemed autocorrelation kind of a nuisance, were focusing on distributions that were very different than what we found in the empirical analysis of the data set. Now, beyond this, autocorrelation has much deeper, much richer, much wider range of structure to it than simply an autocorrelation at a single time lag. Autocorrelation present in animal movement is a rich and multi-scaled process. And so taking advantage of that information rather than trying to hide it has been the central theme of the work that we've been doing. And so that brings me to this here, where the work I'll be describing focuses on a continuous time stochastic process approach to understanding and quantifying animal home ranges. I'll give you an overview of the models that we're working with and how you estimate the parameters of these, and then how you combine them together with other techniques to delineate home ranges. And so this work that I'll be describing has been led by Chris Fleming, a postdoc associated with my lab and with the lab of my former PhD student, Justin Calabrese. And so Chris did his background in statistical mechanics, his PhD at physics at the University of Maryland. And he's very much familiar with non-Markov stochastic processes and the consequences of that structure. And so what he did was to develop the initial derivations that allowed us to start asking questions of the kinds of data that we had from a work championed by another previous PhD student of mine named Thomas Mueller. And so it had to do with the movement ecology of Mongolian gazelles. Here you see one here outfitted with a GPS collar in the grasslands of eastern Mongolia. And the idea is that central to this paper in the American Naturalist is the idea that animal tracking data can be analyzed using a continuous time stochastic process. You can estimate the parameters of that underlying process from GPS location data. And the idea through all of this is you're trying to approximate the movement process that would give rise to the complicated movement tracking data that you have available. And then having made that link between the data and the underlying process, use the parameterized model to estimate long-term space use. And so there are a variety of continuous time stochastic processes that you may be familiar with in mathematical biology. Just as an overview, this is a comparison between individual level stochastic processes and the collective level representation of several different such models. So for example, Brownian motion at the individual level corresponds to a diffusion process where the mean squared displacement is going to be linear as a function of time. In physics, there's a thing called generalized Brownian motion, which leads to what is called anomalous diffusion, where the mean squared displacement is proportional to a power law of time. There's language like subdiffusive and superdiffusive that shows up in this same context. The generalized Brownian motion framework can be developed from continuous time random walks. It's sometimes called fractional Brownian motion. It actually has relationships to these mathematical curiosities called fractional derivatives, which I don't pretend to know very much about, but a lot of mathematicians seem to be unduly interested in them. But those are probably the most familiar. This is very heavily interested in physics, for example. It also connects to things like levy flights. But there's a more complicated form called Ornstein-Uhlenbeck motion. And this is basically a diffusion process with a central tendency. And so what the Ornstein-Uhlenbeck process does is it creates autocorrelation in the position at which individuals are located over time. And that autocorrelation decays as an exponential function of time. And so you end up with mean squared displacement which saturates as a function of time, as opposed to these two, which will continue to grow as time gets bigger and bigger. So if you look at the underlying mathematical structure, Ornstein-Uhlenbeck motion can be described by a one-dimensional Langevin equation here. It's more complicated in 2 dimensions, but this is a stochastic differential equation framework where you have an attractive center that is the location to which an individual is being tended to be pulled back to, like it could be a den or some kind of central attracting place within the animal's home range. There's a time scale for the position autocorrelation, and then there's a white noise process. And this white noise process is uncorrelated in the OU model. And then what Chris derived for the first time in this paper in American Naturalist is what we call Ornstein-Uhlenbeck motion with foraging, or OUF. It is a doubly stochastic Langevin equation where you are now allowing the model to have autocorrelation in both position and autocorrelation in velocity, both of which decay exponentially with time, but they can decay over different time scales. And when you do this, you have a diffusion process with this central tendency, but it's more complex. And so mean squared displacement is now sigmoidal as a function of time. And so it has an additional parameter in addition to the tau h for the autocorrelation of position. You now have an autocorrelation for velocity, which we call in this ecological paper tau f for foraging. We now have a white noise process which modifies the uncorrelated white noise process in this equation here for the velocity, which enters into the equation for the position there. So it's a more complicated version of this otherwise well-studied stochastic differential equation. And so in order to work with these kinds of models, the framework for parameter estimation is based upon what's called a semi-variogram. And so a semi-variogram fit, and I'm going to apologize here to geostatisticians or econometricists who are working with similar data, but it looks like this. Your y-axis is something called a semivariance, and the x-axis here is, in our case, the time lag between a pair of points. And so in the y-axis here, you're looking at how far apart individuals are as a function of these different points in time. And you're doing this for all possible pairs of points. Now, if you would like to trick your graduate students during their preliminary exams, you can ask the question, where does the half come from? Why is it called semivariance? Does anybody have an idea? Okay. This does create the deer in the headlights look when the graduate students are asked this question, but it is very simple because when you have a collection of data points and you're trying to calculate a variance across all pairwise combinations, you could take position A as A, or you could take it as B, right? And so it's simply you divide by 2 because of that reason. It's very simple, but it tricks up everybody. So the idea is that you have a curve that looks like this. It's used all the time in geostatistics and econometrics. And there are relationships between other ways of characterizing the structure of models like this. There's relationships between semivariogram, things called autocorrelogram, and periodogram, and you can make relationships between these. The issue is that for the purposes of parameter estimation, we have found the semivariance approach to be much better as a statistical target for understanding the parameters that drive the process. When you do a parameter estimation by a semivariogram fit, here's an example of one particular Mongolian gazelle. So this is its movement track. This is 250 kilometers or more on this axis and 150 kilometers or more on this axis. This breaks it down by the X, Y, and coordinates separately. And you can see this animal is not, over the course of 1 year, following a regular pattern of motion like you might expect in a migratory species, or following a pattern of motion that's keeping it clustered in a particular area. It's wandering all over the place. But yet you have to ask the question, is it just going to continue to wander forever, or are there going to be some bound? And so you can look at the data shown here in blue, and you can see that there's definitely a tendency for the semivariance to saturate as you get to very long time lags. There's many fewer points out here at the long time lags. There's many points at short temporal time lags, because when you look at your data set, you have lots more points that are close together than you do that are much farther apart. And so the criticality to estimating parameters of a model like this, it depends on the entire multi-scale process here across all these lags, but there's an extra abundance of points at these short time lags that can play a key role. And so you can see the Brownian motion was a bad fit, because that's when you go linear. This thing is clearly saturating. The anomalous diffusion, again, linear on a log scale, that's not going to help us, but it's postulated as a reasonable explanation for a large scale movement process. It doesn't work for the Mongolian gazelle whatsoever. The best fit Ornstein-Uhlenbeck model shows the kind of saturating structure that we see in the blue empirical data, but really you can see it's missing a lot. It looks visually like it might be onto something, but it's not enough. And so that's how Chris decided to derive this OU plus foraging model, which provides a much better fit. You can look here, the AIC values. The difference between the OUF and the next best performing model is hundreds of AIC units. So this is clearly a very strong match compared to the alternatives. But if we blow up now to see what's going on at this small scale, you can see the sigmoidal structure. So here's the data in blue, in OUF is in red, the standard OU model is in green. This OU is saturating, the OUF shows this concavity here. And that concavity is due to the fact that you have autocorrelation in position as well as velocity in the OUF model. And that induces a sigmoidal shape in the semivariance function, which is important to capture when trying to make predictions about space use. The model is a much better fit to this kind of stochastic process than to one that doesn't have this extra level of complication in it. And not only does it work in this particular Mongolian gazelle, it has it works for a wide variety of different individuals thanks to a sequence of methods that have been developed over the years to facilitate fitting variograms to weak, messy, incomplete, just generally nasty data sets that you might get from a real ecological time series of an animal movement. These are summarized in a Methods in Ecology and Evolution paper led by Inez Silva in 2022. They start from very simple and they end up with something as complicated as bootstrapped perturbative hybrid residual maximum likelihood techniques, which just rolls off the tongue. But what that means is that it is structured so as to remove as many different kinds of biases in the estimation process as we could think of for very weak data sets. And so this is a challenging way of fitting this. It is very time consuming for some data sets, but it will help in the cases where the data are messed up pretty badly. It will work. It works up until certain circumstances when the data simply become too weak to estimate these kinds of parameters. And those are discussed in length in this summary paper by Inez Silva and colleagues. Now, when we looked at this across about 2,000 mammalian movement tracks now, 80 to 90% of those GPS tracks that we have fit using these approaches are OUF. The OU model, or an alternative, even simpler model called an IID model, which will give you a saturating variogram. They're only found in rare cases, and they're only found typically for very low frequency sampling. In modern days, when you have a good quality GPS recording device, you're getting lots of data on fine scale, like minutes to hours, you will pick up this kind of autocorrelation in velocity. And this is work that we discussed in Mike's monograph and also in this paper, which I'm calling In Eternal Review, which I'll be talking about on Friday, where we've done this for lots and lots of carnivores as well. And so when you do this parameter estimation by semi-variogram fits, you get what are called key scales of motion. They are the parameters that govern the time scale of relaxation of the position and also the time scale of relaxation of the velocity. And those correspond to spatial scales over which that relaxation in the autocorrelation plays out. And so biologists' audience, we call these tau F, tau H for the two different time scales, and then LF and LH for the two different length scales over which these processes play out. But they correspond, perhaps in mathematical world, or definitely in the physicist world, to other scales which are more naturally described in those settings. And so there's a correspondence.
Maybe you want to talk about it at the end, and maybe Farouk can ask.
Okay, yeah.
Okay.
All right, so the question was about non-stationarity versus stationary, and I can't address that. But so this idea here is that we can characterize these timescales over which the velocity and the position are autocorrelated, and over which the length scales for the same correlations. And then you can go a step further. This thing called LV, which we call the length scale over which the animal's velocity is autocorrelated. The physicists call this the ballistic length scale. And what this corresponds to, if you think about it in verbal terms, this is the average distance over which an individual will show persistence in direction. There's an equation that governs this ballistic length scale. You can calculate this from the ratio of the two temporal scales times this thing called sigma p, which is defined as the spatial variance in the animal's movement. Now, you might think that since this is a variance, it should show sigma squared. This is not my fault, even though I am a biologist and I'm prone to mathematical mistakes. This is actually something that has to go back with where the process was originally derived decades ago. And the people that did that work used sigma without the square to call it a variance. So I'm just doing the same thing. I'm taking this sigma sub p, it's in the side of the square root. And so by taking the square root, you end up treating it as a standard deviation, okay? It makes sense. The units work out. It just looks funny and it sounds weird when you talk about it that way, because a statistician will say the variance, it has to have the square, sigma squared, right? Doesn't work out when the inverse derivation. But this is, it's the spatial variance in the animal's movement. It's relatable to the white noise that's present in the OU and OUF stochastic processes. And this tau v, this temporal scale over which the velocity is autocorrelated, it only appears in OUF movement. There's going to be more on this on Friday. I'll talk about ballistic length scales and how they might be useful in interpreting animal spatial and maybe even some conservation biology questions, but you have to come to the other seminar to get that. Okay, so the idea is that when having estimated these parameters from these stochastic process models, the next step is to actually put them into this framework that I mentioned earlier called kernel density estimation. And so kernel density estimation, as I was showing, it works. You start from an array of points and you put small kernels of this bandwidth or covariance located at each sample point. And then you take a smoothing, you take a spatial average to obtain the probability density function of that process. And you end up with the estimated probability density function, which corresponds not just to the bounds of the animal's home range, but actually shows its lumpy structure, the areas that are going to be more heavily used versus less heavily used. The trouble is that conventional kernel density estimation assumes that the sample points, the things that you've collected, are IID. And that is a problem because, as I said, animal movement tracks are highly, highly autocorrelated. And so that requires a change. It requires that we move to autocorrelated kernel density estimation, which means instead of having a uniform bandwidth here, you replace those with optimal kernels whose optimal values are shaped by the autocorrelation structure in the track. These are things that are calculable from the semivariogram. Those parameters that we went to great pains to calculate now come back in to shape the kernel that we're going to use in this KDE. And it also weighted for the specific distribution of points in the trajectory, which doesn't have to be regular, right? A biologist might program a collar so it takes a series of points in short succession and then has a gap, followed by another series of points, followed by another gap. The data don't have to be regular. And so in order to work with those kinds of data, you have to be able to deal with the irregularities in them. And one way of doing that is to develop an optimal weighting scheme, which has been developed and implemented in this series of papers here. Again, lots of work led by Chris Fleming, who's now an assistant professor in biology at the University of Central Florida. And it makes a big difference when you account for autocorrelation. So here's an example. This is for one real gazelle track, the one I was showing you earlier. On the left, we have the kernel density estimate from a home range for an animal studied for almost a year. This model assumes that the data points are spatially independent. You can see that the home range that you get out of this process has this little island up here. It doesn't capture this trajectory here where it's moving between the island and the mainland of the home range. And the AKDE, on the other hand, has a very different prediction about what this animal's home range should look like. This is a prediction of its asymptotic home range. That is, if the animal were to show the same kinds of behavior over the long term, what space would it exhibit? And so that is what is being shown here. And you can plug this in and you get not just the average home range, this is the 95% quantile of that home range, you also get confidence bounds on the location of that 95% quantile. So this AKDE accounts for autocorrelation in movement data. There's an additional advantage in that we can, because of the way that the kernels are optimized and then weighted as part of this process, we can make this work with weaker data sets. So let's take the same gazelle, cut the data in half, and let's see what we get for predicting the home range based now on only 180 days worth of data. Well, interestingly, the animal's second 180 days, that's when it took this big sojourn in orange. That's not part of it happened, in the second 180 days. It wasn't part of the original data set. And so when you plug this shorter time series into the kernel density estimation, you get a home range which is substantially smaller than this one, and it's much more fragmented with more boundary and less core area. When you do the same thing, again, working with only half the data, and now you put it into the AKDE estimator, you find that the prediction is very similar to what you saw based upon the full data. And in fact, the autocorrelation structure that you were able to estimate that being present in the animal's trajectory based on the first 180 days contained enough information about how it might be likely to move that it could accommodate this idea of this big time wandering loop that took the animal away from the area that it had been seen in previously. And so the autocorrelated kernel density estimation based on the first half of the data gave a very good approximation to what we saw in reality downstream as we gathered more information about this individual. So AKD offers better predictions and is okay with fewer data. It has a clear benefit compared especially to techniques like minimum convex polygon and other approaches that embrace and take advantage of and use the mathematical power of the autocorrelation rather than eliminating. So it has real virtues. I want to make a final pitch here before going on to the last section of the talk about what we're actually estimating in doing this kind of AKDE kernel density estimation. The kernel density estimation is a range distribution. Asymptotically, what we are predicting is where the animal will go, what space it will use. That is very different from another very popular set of tools called the Brownian bridge density estimation, which is based upon the idea of a Brownian bridge or any kind of stochastic bridge between points in a sample landscape. This kind of estimator, when you look at it statistically, what it's yielding is an occurrence distribution, which is fundamentally different from a range distribution by several different factors, which I want to go over just so you understand the differences and why there's a big concern, because this technique was developed before this technique. And there's a lot of people that have invested a lot of time in this thing, but it's yielding a different distribution. So for example, the AKDE is a range distribution and it measures the intensity of space use. Statistically, it's obtained as a marginal distribution. On the other hand, an occurrence distribution measures the uncertainty in spatial position. And it is based upon a conditional distribution, conditional on the actual sample points. It is not a marginal distribution. The consequence of this is that the AKDE lends itself to questions of extrapolation, looking and projecting over time. Whereas the occurrence distribution based on Brownian bridge or even OU bridges, other kinds of stochastic bridging approaches, are fundamentally an interpolation technique. That means that you can fill in the blanks and you'll get information about what happened in between those sample points, but you cannot extrapolate. You're doing an interpolation. So the AKDE does not exhibit sampling dependence. The Brownian bridge techniques will exhibit tremendous level of sampling dependence. And in fact, if you were to have a movement track that was so perfect that you were getting, second level data for days and days and days, and you applied a Brownian bridge technique to that, you would recapitulate your estimate of the so-called home range that you would get from this technique is simply you would recapitulate the movement track itself. It is narrowly defined based upon that sampling dependence. If you have a coarse sampling, you will estimate much broader space use, but that is only because you are estimating uncertainty in spatial position. John.
In what sense do you say that the APD doesn't have sampling dependence though?
It
I mean,
doesn't
it
have,
seems that it still builds from sampling, so in what sense is it not have dependence?
in the sense that, if you have, very coarse scale data separated by, as long as you can estimate the underlying parameters, which is constrained by the set of techniques I was talking about in the syllabus paper.
Yeah.
As long as you can estimate the underlying parameters, you will not get a prediction which is based upon how coarsely those points are sampled apart. That's what I mean. You can do the same thing that I did here, where I cut the data in half. You can do the same thing based upon thinning. And if you were to thin the data, you would find the same idea, that down to a certain threshold beyond which you can't reconstruct the variogram. As long as you don't thin beyond that point.
Guess that's what I'm getting at. There has to be a threshold.
Yeah, it's
You
like,
can't recover
it's
the,
like if you cannot estimate the underlying velocity correlation or the underlying position autocorrelation, you're going to have problems. But you can apply this technique to very coarsely data that are treatable as if they were IID.
okay, fair enough.
And so the point I want to make is both techniques are useful, but they should be used for different questions. So if you want to ask how large is an animal's home range, or how do environmental and social factors influence an animal's area requirement, you want to use a range estimator. And your product you want to have is a range distribution. If you want to ask instead, how likely is it that an animal has visited a particular location of interest, or where might an animal have crossed a linear feature, then you want to use an occurrence distribution based on a Brownian bridge or OU bridge or something like that. These points are made mathematically in the appendix of this paper, which not a lot of people have read, but we have written this other paper led by Jesse Elston, who's now at the University of Arizona, which is in revision, second revision at Ecology, but it's available in bioRxiv and ours been cited a bunch of times. But the idea is this framework, this dichotomy is very well expressed in English in this paper for users, even though this one is based on the mathematics. It gives appeals to a more limited audience. All right, so AKD offers improvements. Does it actually matter? And so here's the size of Yellowstone. Here's the total protected area in Mongolia, all national parks, 15,000 square kilometers. Here's the AKD estimate of the average gazelle home range taken across individuals. That's 75,000 square kilometers. These animals are moving, and you can't estimate this without having the opportunity to understand just how big an area they will use over their lifetime. That's one animal. That is a huge expanse. It has to, think about the consequences of that for conservation planning. That's equal to the area of Panama. So I mean, the situation is only going to get compounding as the autocorrelation is more and gets stronger and stronger in the data that are being collected. All right, so I want to give a quick overview in just a couple minutes here of other applications and some recent advances that we've done to take this foundational idea and extend it in a couple areas. One of these has to do with learning and the establishment of space use by an individual as it's getting information about its landscape. And the other has to do with population level attributes. In a paper with Mark a couple of years ago, we looked at how learning can be reflected in animal movement tracking. And one of the ideas there was that as an animal gets information about its environment as a process of exploration, that can change how it uses space to a more exploitative relationship. And so we might be able to pick that up where something like a metric like the probability of revisiting a particular location could depend upon time since translocation. And these other ways in which learning can also change and be reflected in animal movement signatures. And so we wanted to extend this idea. And so we looked at other systems where people have studied learning. For example, there's learning of flight routes by homing pigeons, some really cool ideas about how animals' movement paths become fixed over time, so that by the time an animal is trained, it ends up taking a very long detour. It doesn't take the most optimal route back home. It can have a very long detour as part of what it has learned about how to solve a particular navigation problem. That's from Dora Biro and her colleagues. My student Thomas Mueller looked at social learning and whooping cranes, found that the presence of other individuals that have more experience can shape the movement ecology and migratory efficiency of younger birds that happen to fly with those older birds. The idea then is, stemming from this work on the whooping cranes, which were raised in captivity, we wanted to ask, is there information that might be available about how animals that are newly released from captivity might be behaving? Typically, there's going to be some kind of a disturbance from the release process, but that's something that's going to be on a smaller time scale. Barring some kind of injury, it's going to be a matter of days or even a few hours as the animal adjusts to being out of captivity and in the wild. And there's different techniques that have been employed to try to deal with that so-called capture or handling effect. There's a larger issue of an animal which is naive and being thrust into a new environment where it has to gain information. It's unfamiliar with the location in which it's been located, and this is potentially much longer lasting. It represents an experience deficit which can only be overcome as the animal gets more information about it. And so a key question then is, how long does it take for naive animals that are released into new environments to gain experience and information about their surroundings? And so we want to quantify experience gained as evident in a change in behavior. There's a variety of approaches that can be employed to look at this that have to do with different kinds of information that are gathered as animals are released. So here's one such study. This is something led by the Smithsonian and other groups to reintroduce a species called the scimitar horned oryx, which was extinct in the wild. And so this is the release process of a group of oryx, which has been raised in captivity, being released into this national park in the middle of the country of Chad. And this is one example of how this release process might work. Another example is what happens with the Sonoran pronghorn, where these animals do not like captivity, they do not like people, and basically the best way to keep the animal from injuring itself is to have a gang of people hold the animal and move it from one place to another and then let it go. It is a much more challenging process than you see here, where these oryx are just happy to be sitting inside their boxes. This is where you hope you don't get injured during the process. And then, of course, there's other kinds of approaches, such as here, where a panda is being released from captivity by two people that are dressed up like pandas. This is not how this operated initially. When they first released pandas into the wild, it was a media spectacle event in which everybody and their brother was there, and they had camera lights and everything going on. So it's become much more sedentary and sedate as time has gone on. But there's a variety of methods that are used as animals are released into the wild. And the question is, are there any echoes of this in the movement data? The commonality for all of this work is that you have naive individuals that are being released into new environments. And that experience gain is something we expect to map onto a change in behavior. So when you're doing this from a statistical standpoint, what you're talking about is a change point detection technique, which is something very common in time series analysis, where people, depending upon what they're trying to characterize, are looking at changes in means, they're looking at changes in standard deviation, or other ways in which the time series could be analyzed. The idea is, can you identify when the signal has changed? And we can do the same thing with animal movement data. And so one of these, some of the earliest work on this was led by my former postdoc, Ellie Garari, who's now at SUNY ESF in New York, what he called behavioral change point analysis. It started with a paper in Ecology Letters. More recently, he's developed this package called S-Move for correlated velocity models like those that we use in the autocorrelation analyses. The idea is that you can look at changes in behavior. This is the log of some movement metric, whether it's one of these parameters or a displacement metric or something else, as a function of time since the animal was released into the environment. And so Mark may not remember this, but he's a co-author on this paper on pandas, in which I was invited to China, I think in response to the fact that we had the cover of Science on the whooping crane paper. They wanted to get a cover of Science for their panda paper as well. And they had data for us, but they had 2 individuals. And so I can work with a lot of messy data, but I cannot get a cover of Science on 2 individuals. And so we are stuck with a paper in Ursus, which is the journal of the bear researchers of the world. But the idea is we can look for these kind of signals in the animal data as the animal was released from captivity. And so here's data for 2 different giant pandas. And you can see that initially the animal stays in a very small area. And then you can see an echo, or you can see an indication of a change in its behavior as it started to move through the landscape a little bit more, it broadened out. And then over time, it started to make much broader forays into the landscape. The same thing happened for a different panda here. They have long-term transition periods of multiple months in both cases that are evident in the movement track as they've gained as they explored more extensively. In the Sonoran pronghorn system, my student Nikki Barber, who's now on the faculty at Towson University in Maryland, took a similar idea. She wanted to ask what happens to the speed of the animal after they've been released. So after the change point, she found that some animals will slow down after the change point. Others will speed up. And having identified the distribution of changes in speed shown here, she found about 22 of the individuals decreased in speed after their change point and 36 increased in speed. She had a much larger sample size than the 2 pandas that we had to work with, but she found that the transition point was about 47 days. Some of them seemed to adjust after only 17 days. Some took much larger. But she was able to distinguish between animals that would eventually become range resident and animals that would continue to be nomadic wanderers for the duration of the information that we had on them, which in most cases was several years. And so the nomads were the ones that sped up after this transition point. So they walked around at a given pace after their initial release. But then if they were, in times later, if they sped up after that period, the initial period after the release, they would eventually continue to wander for the duration of the information that we had on them. They would not settle down. Whereas the range resident individuals were those that either slowed down immediately after their release or exhibited small scale increases in speed. She was able to find differences between males and females, where the males tended to be the ones that were nomadic, but there's also a group of males which slowed down tremendously in speed and became range resident, in a sense, territory holding individuals. And so we've got this distinction, these 2 different male strategies that emerged, range residents, which had short transition periods followed by a decreased speed, versus persistent wanderers that were long transition periods followed by increased speed. And so that is her summary of that work. And then the ongoing project with the oryx, we have much bigger data set, 247 GPS animals. And this is work that's being led by my student Gayatri Anand. And she's, because of the scope of this data set, we're able to ask questions about whether this transition period following the release depends upon group size or total population size, or even the methods by which the animals were released. And so this is something we're looking forward to working on with this accumulated data on oryx. And so I'm going to stop there because I've gone long and it's not going to get to the population level work. But the basic idea is that there's 2 related applications. One is that we can take the AKD methods and we can scale up the inference, so we can now ask questions at the population level about individual range distributions. And we can also scale up in a different way to take individual samples and make a prediction about how big the range of a population of sampled animals will be. So these are 2 different ways in which we can scale up from these individual observations to population level information. So at that, I'll stop and be happy to take any questions.